Okay, ready to dive deep into something truly mind-blowing.
Absolutely.
Today we're tackling microRNA,
Interesting.
tiny bits of genetic material that have a huge impact on how our bodies work.
It's fascinating how something so small can be so powerful.
I know, right?
Yeah.
It's like the saying dynamite comes in small packages,
Mhm.
but instead of explosions, we're talking about controlling what goes on inside our cells.
Precisely. You see, for a long time, scientists thought they had gene regulation all figured out.
Really?
Yeah, they knew about DNA, our genetic blueprint, and how it gets transcribed into messenger RNA, the instructions for making proteins. They even knew about transcription factors,
Okay.
those proteins that act like switches turning genes on or
Like
off.
flipping through that giant instruction manual and deciding which pages get read.
Exactly. But then two scientists, Victor Ambros and Gary Ruvkun, came along and totally changed the game.
Oh, really?
Oh, yeah, they were studying these tiny worms called C. elegans.
C. elegans.
Think of them as the lab superstars of the genetics world.
Okay, so what were these little worms up to that got scientists so excited?
Well, Ambros and Ruvkun were looking at these mutant worms that had some really strange developmental issues.
Hmm.
You know, growing body parts in the wrong places.
Okay, it's more than a little strange. Like growing an extra nose where your ear should be.
Something like that. It was clear that something was messing with the worm's genetic instructions, causing them to develop incorrectly.
Wow.
They zeroed in on two genes, LIN4 and LIN14, that seemed to be at the heart of this developmental drama.
So LIN4 and LIN14 were the masterminds behind the misplaced body parts. What did they find?
This is where it gets really interesting. Ambrose discovered that LIN4 was producing this super short RNA molecule that didn't seem to code for any protein.
Huh.
It was a real head-scratcher. What was this little piece of RNA doing?
It's like finding a random note in that giant instruction manual, and you're like, what's this all about? Does this even belong here?
Exactly.
Yeah.
Meanwhile, Ruvkun was busy investigating LIN14, and he found something equally perplexing. He realized it wasn't the production of the messenger RNA that was the issue. The instructions were still there, but the protein itself wasn't being made.
Hold on. It's like having the recipe, but someone keeps swiping the cake out of the oven before it's even baked.
That's a fantastic analogy. It was as if something was interfering with the protein production process.
Okay, I'm hooked. We've got a mysterious short RNA and a case of disappearing proteins. Did Ambros and Ruvkun ever figure out what was going on?
You bet they did. And what they found would rewrite our understanding of how genes are regulated. It turned out that Ambrose's little piece of RNA from LIN4 was binding to the LIN14 messenger RNA, preventing it from being translated into a protein.
No way. So this tiny piece of RNA was basically putting a stop to the protein production.
Yes.
It's like slapping a do not make sign right on those genetic instructions.
Precisely. They had stumbled upon a whole new level of control.
Wow.
A way for cells to fine-tune gene expression using these tiny RNA molecules. They called these molecules MicroRNAs. And they're anything but micro in their impact.
Wow, microRNAs. They're like the tiny but mighty guardians of the genetic world.
Yeah.
But hold on. If these microRNAs are the heroes of our story, how do they actually work their magic? I mean, do they just float around the cell until they bump into a matching messenger RNA? I'm guessing it's a little more complicated than that.
You're right. It's definitely not random. MicroRNAs go on a whole journey, a carefully orchestrated molecular dance to find their targets and exert their control. Think of it like a micro-odyssey with multiple checkpoints along the way.
A micro-odyssey.
Well, yeah.
I love it. Okay, I'm ready to embark on this adventure. Lead the way.
All right, so our story begins in the nucleus, the cell's control center, where the microRNA gene is first transcribed into a long RNA molecule. This is like a rough draft, if you will.
So it's not ready for prime time just yet? What happens next?
This long RNA then folds into a specific hairpin-like structure. Imagine taking a long piece of string and doubling it over itself.
Got it. We've got our hairpin RNA. Is this when it gets its microRNA makeover?
Exactly. This hairpin RNA gets trimmed down in the nucleus by a protein aptly named drosha.
Drosha, is it like a tiny molecular stylist shaping up that RNA into a chic microRNA?
You could say that. This trimmed-down RNA, now called pre-microRNA, is then escorted out of the nucleus into the cytoplasm where the real action happens.
Out into the bustling city of the cell. What awaits our pre-microRNA in this cellular metropolis?
In the cytoplasm, our pre-microRNA encounters another key player, a protein called dicer.
Dicer, okay, that name makes sense. I'm picturing tiny little molecular scissors snipping away.
You got it. Dicer does some more snipping and dicing, hence the name, to create a very short double-stranded RNA molecule.
So we've gone from a long single strand of RNA to a much shorter double-stranded one. That seems like a lot of effort. Why not just make the short one to begin with?
That's a great question. This multi-step process is actually a form of quality control. Each step ensures that only the correct microRNAs are produced and that they're the right size and shape to do their job.
Ah, so it's like a microRNA assembly line with each step ensuring the final product is perfect. But we've still got a double-stranded RNA, right? Doesn't a microRNA need to be single-stranded to do its job?
You're one step ahead of me. Yes, one strand of this short double-stranded RNA is selected as the guide strand. That's the one that will be our little puppeteer.
And the other strand, does it just get tossed aside like a rejected dance partner?
Pretty much. The guide strand then gets loaded onto a protein complex called RISC, which stands for RNA-induced silencing complex.
RISC. Catchy name.
All right, so our microRNA guide strand is all dressed up with its RISC complex escort. What happens next in this microRNA odyssey?
This is where things get really exciting. The RISC complex uses the guide strand like a homing beacon, scanning other messenger RNA molecules in the cytoplasm for a complementary sequence.
So it's like trying to find the matching puzzle piece.
You got it. When the RISC complex, guided by the microRNA, finds a messenger RNA molecule with a matching sequence, usually in a region called the 3' UTR, it latches on. Okay, our microRNA has finally found its target. Now what? Does it just give it a high five and move on?
Not quite. This is where the microRNA, as part of the RSC complex, flexes its regulatory muscles. It has two main options. It can either block the messenger RNA from being translated into a protein, essentially silencing that gene.
So it's like putting a big do not use sign on those genetic instructions.
Exactly. Or it can direct the messenger RNA to be destroyed, getting rid of the instructions altogether.
Wow, so microRNAs have the power to completely shut down a gene.
That's right. They are incredibly powerful regulators of gene expression, and this is where their importance really comes into play. By controlling which proteins are made and in what quantities, microRNAs influence a wide range of cellular processes.
Okay, I have to ask, this all sounds incredibly precise, like a well-choreographed ballet of molecules.
Yeah.
Is it possible for this whole microRNA system to ever go wrong? What happens if our tiny dancers miss a step?
That's a great question. Like any intricate dance, things can go wrong.
Yeah.
If microRNAs aren't produced in the right amounts, or if their timing is off, it can disrupt the entire cellular performance.
So it really is like our conductor suddenly deciding to speed up the tempo or cue the wrong section. Chaos in the orchestra. But what does that kind of chaos mean in the context of our health?
Well, you can imagine if a microRNA that's supposed to help regulate cell growth suddenly goes missing or becomes overzealous.
That sounds like a recipe for trouble.
Exactly, and that's what we see in certain diseases, including cancer. In some cases, microRNAs that normally suppress tumor growth are absent, allowing cells to multiply uncontrollably.
So these tiny molecules can have a big impact on something as serious as cancer. That's incredible and a little scary, if I'm being honest.
It highlights just how important microRNAs are for maintaining our health.
Yeah.
But here's the good news. This also presents an incredible opportunity for developing new treatments.
Wait, are you saying we could actually use microRNAs to fight diseases like cancer? Tell me more.
Imagine being able to target the very root of a disease at the genetic level. Instead of just treating the symptoms, we could potentially correct the faulty gene expression that's causing the problem in the first place.
So instead of just trying to mop up the mess, we could prevent the mess from happening in the first place. That's a game changer. How would that even work?
Let's say we know a specific microRNA is underproduced in a certain type of cancer, preventing it from doing its job of keeping cell growth in check.
Okay, so our tiny conductor is missing from the orchestra pit.
Precisely. In that case, we could potentially deliver synthetic versions of that microRNA directly to the cancerous cells.
Essentially giving those cells a much-needed tune-up.
I love it. But are scientists actually working on this kind of microRNA therapy?
Absolutely. It's a rapidly growing field of research with enormous potential jobs. Scientists are exploring ways to use microRNAs as both diagnostic tools to identify early signs of disease and therapeutic agents to correct the underlying genetic imbalances. This is mind-blowing stuff. We've gone from tiny worms to potentially revolutionizing medicine. It sounds like the future of healthcare could be very micro.
I like that. It's important to remember that microRNA research is still in its early stages, and there are many challenges to overcome.
Right.
Delivering these tiny molecules to the right cells in the body without causing unintended side effects is no easy feat.
It's like trying to deliver a tiny package to a specific address in the middle of a bustling city. But even with those challenges, the possibilities are incredibly exciting.
Absolutely. Who knows what other secrets these tiny molecules hold? There's still so much to learn and explore in the fascinating world of microRNA.
Well, you've definitely left me wanting to learn more. It's amazing to think that something so small can have such a huge impact on our lives. It makes you realize that there's a whole universe happening inside each and every one of us, orchestrating this symphony of life. And for you, our intrepid listeners, we leave you with this thought. If you had the power to control the expression of any gene in your body with microRNA, which gene would you choose and why? The possibilities are truly endless. Until next time, keep those minds curious and those questions coming.